Hi, this is Miss Litton, and this is my wonderful 7th period honors bio group. Say hi.
Hi.
All right, so this was all about Mendelian genetics. So I'm going to hit the big ticket items, and then we'll do a bunch of practice problems, and we'll look at some pedigrees. Does that sound good? Okay, so the first thing I want you to make sure you remember are the 3 laws, okay? Do you remember the 3 laws? Yes?
Independent Assortment.
That's one of them. Okay, so the first law is the law of dominance. That means when you have a big R and a little r, that particular pea plant will be round, right? In order to be wrinkled, what would you need? Two little r's, okay? So that's the law of dominance, is that one allele would be expressed over another allele. Now, if you're taking notes on this, let's talk about exceptions to the law of dominance. Exceptions to the law of dominance. So, can you think of some?
With blood types like IA and IB.
I love it. And that, so exception number 1 we could say is codominance. That's when both alleles are expressed equally, as in AB blood. Okay? The A antigen is expressed and the B antigen is expressed. I'm trying to find it. Here it is. Okay? So this right here is one exception to that rule. A is not dominant over B. Okay, another exception to the law of dominance.
Isn't it when like it blends together like the
Yes, do you remember what we call that?
Crossing over?
No, incomplete dominance, right? So somebody who has straight hair reproduces with somebody who is homozygous for curly. So homozygous straight, homozygous curly. They make a baby. That baby is H1, H2. It looks like blending to our eyes. It looks like blending. But it's wavy. It's like neither. And then another example would be a red flower crossed with a white flower, and it's a pink flower. A black bunny crossed with a white bunny, and they have gray hair. But if you crossed a black bunny with a white bunny and the baby bunny had some black hairs and some white hairs, we would call that codominance. Truth be told, these two are actually probably one and the same, right? Because the reason why it would appear gray, right, is because the black and the white is expressed. You understand that? So it's really probably the same thing. Okay, so we have codominance as an exception to the law of dominance. Incomplete dominance. Can you think of something else? Yes?
Is it law of segregation?
No, that's the one law we are not. That's our next law. I'm talking about exceptions to the law of dominance. Exception. How about for my gentleman in the room? You only have one X. So we're not going to be, if you were a female, you have 2 X chromosomes, so you will have 2 copies, 2 cards to play if you're a female. 1 X you got from your mother and 1 X you got from your father. So you can play both those cards. One allele may be dominant over another allele. Okay? It does not work that way for males because they have only 1 card to play. And who gave them that card to play? Their mom gave them that card to play. That's the only card. So it doesn't matter if the allele is dominant or recessive, they're playing that card. So if it's a disease, right? And if it's a recessive disease, normally you would need 2 bad copies, which would mean mom would have to give you a bad copy and your dad would have to give you a copy for you to get that recessive disease, right? You agree with that? But in this case, you only need 1 bad copy if it's on the X chromosome because gentlemen only have 1 X chromosome. Okay? So that would be another exception to the rule. Codominance, incomplete dominance, sex-linked traits for males. What's another one? Polygenic. Remember polygenic traits? So it's not like, we're not like pea plants where you're either tall or you are short, right? We have varying heights. That's because it's not big T, and you're a really tall person, big T, you're a medium person, and little t, you're a midget. It doesn't work that way for humans. Okay? We have several genes that contribute to our height or our skin color, our intelligence, and we call that a polygenic trait, and they're all working on that one trait. So that's not a straight up dominant recessive type of thing. Okay, and if you recall, I told you those polygenic traits, especially those disorders, are more susceptible to environmental influences than other straight up traits. Okay, and when we looked at environmental influences on traits, we looked at like when you shave the back of a Himalayan rabbit and when you put an ice pack on there, because that's exactly what that Himalayan rabbit wants, that when the hair does grow back, it grows back black. Why? It was exposed to cold, and black hair can absorb more light, right? So that's a response to its environment. Does that make sense? It's a response to its environment. And the same thing has to do with alligator babies or reptile babies, that oftentimes the temperature of the nest will determine the sex of the offspring. And that brings us back to our nature versus nurture environment. How much of our phenotype is determined by what's in our genes, and how much of it is influenced by our environment. Okay, so those were the laws. That was the law of dominance and the exceptions to that law. Now, what was the second law that we discussed?
Segregation.
Yes, law of segregation. Now, the law of segregation says, okay, that when you have a homologous pair of chromosomes, that they will segregate during meiosis 1. You'll separate the homologous pairs. And then in meiosis 2, you will separate the sister chromatids. Perfect. Okay? And so that particular law, am I still recording? I am. Continue. That particular law does not get broken in any normal fashion. Okay? So if you fail to separate, that's called nondisjunction, and there will be issues surrounding that. For instance, Down syndrome, okay? And that's trisomy 21, so 3 copies of the GARD gene. So that's not a normal thing. The law of dominance is one that's broken, because there are exceptions to the law of dominance. But the law of segregation, you have no normal exceptions to that rule, okay? And you can see the law of segregation every time you make a Punnett square. Because if you had one parent who was big G, little g, you separate out the potential G's. You are doing the law of segregation every single time you set up a Punnett square. Okay, got that? All right. Now, we've done the law of dominance, the law of segregation. What is our third law that we want to be concerned? Yes, we want to talk about the law of independent assortment. Here we go. Now, the law of independent assortment is specifically to meiosis 1, and particularly what stage?
Metaphase.
Yes, metaphase 1, when the homologous pairs line up in the middle. Each homologous pair, so how many homologous pairs do we have in our cells when we're getting ready to make sperm or egg? 23 homologous pairs. All 23 homologous pairs line up independently of the other. It's not like all the dad ones you got from your dad are on the left and all the ones you got from your mom are on the right. No, they all line up independently. And when we looked at that, we looked at this example and we said, what if these homologous pairs lined up in this way? If they did, then the type of, let's say this person is making sperm, okay? Then the type of sperm you would make is you could get little b with big E, or what's another type of sperm you could make?
Big B, little e.
Big B, little e. But what if they did not line up that way? What if they lined up this way? This way. Okay? Then the type of sperm you would get would be little b and what? Little e, and big B, big E. Big B, big E. Those are all the options in sperm. So when you make your Punnett square, you're not making it like this, right? Where we had big T, little t, or anything like that. Your Punnett square now has to deal with all of the potential gametes that you could form when you do that cross, right? And then remember, we discussed shortcuts to that process. Even if you have a cross you need to do that is this. In fact, let's do this cross right now. okay? This is who I'm crossing. I want to know how many of them will turn out this way. That's what I want to know. How many of them will turn out this way? Okay, and I'm going to pause you just for a minute so they can work. You should work at home too. Sorry, I made a little mistake there. Yeah, sorry about that. My bad. Do the problem. All right, so hopefully you had a time to solve it. So remember, when you have these bigger crosses like this, you do not want to make a ginormous Punnett square. All you have to do is do individual Punnets. So first thing I'm going to do is I'm going to cross my B's. I have a big B, little b, and two little b's. You wouldn't even have to write both little b's, right? It would be the same thing. You with me on that? You could have just written it like this, right? You understand? Because either way it was going to be. So these are your only two options. Okay? Then I'm going to cross my A's. Big A, little a, big A, big A. Okay, and like I said, you get the same results if you only put one of these big A's. Do you understand what I'm saying? Because they're exactly the same, so you can make your Punnett smaller. Okay, and then I'm going to cross very crookedly my R's. And a lot of you can see these Punnets in your head, and it's kind of superfluous to do it. And if you can, then do that, right? Okay, so I'm going to do with the first one. The first one, I wanted big B, little b. So how many big B, what's the ratio? What are the odds I'm going to get a big B, little b? 1/2. Okay, because you always simplify. You wouldn't say 2/4. And then what are the odds of, what did I want next? Big A, little a? 1/2. And what are the odds, and by the way, that's here and here. Okay, and what are the odds of big R, little r? 1/2. So then all I have to do is multiply those together. 1 times 1 times 1 is 1. 2 times 2 is 4 times 2 is 8. Okay, so just remember to multiply and not add. Okay, because remember how we have to do that with probability? You know, if we're 3 best friends and we all go into the hospital at the same time, what were the odds statistically that all 3 of us would have girls? It was a 1/2 chance I'd have a girl, a 1/2 chance you'd have a girl, a 1/2 chance you would have a girl, but that all 3 of us simultaneously had a girl. 1/2 times a 1/2 times a 1/2 is 1/8 chance that would be girl, girl, girl in that order. Does that make sense? That's when you have to multiply it together. Yes?
How do you know when to multiply versus when?
If there's more than one way to get the same thing, that's when you would add it up. So for instance, you can get a hybrid right here. You could say this is big R, little r, and this is little r, big R. There's 2 different ways to do it. So in that case, you would add them and you would say, oh, there's a 1/4 of a chance here, and there's yet another way you could get that same combination, a 1/4 and a 1/4, so that's a 1/2. If I had 2 coins, like you guys were throwing coins at each other earlier, right? If all I said is, I want to flip these 2 coins and I want them to be different from each other. That's all I want. I don't care as long as they're different. Well, this one could be heads and this one could be tails, right? So heads, 1/2 a chance. Tails, 1/2 a chance, right? 1/2 times a 1/2 is a 4. Put a pin in that. Or this one could have been tails and this one could have been a heads. A 1/2 times a 1/2 is a 4. So there were 2 different ways, each worth 1/4, for me to get the same outcome. That's when I add them up. A 1/4 and a 1/4 is a 1/2. It's only of a 1/4 of a chance that I would get head and tail. There's only a 1/4 of a chance that I would get tail and head. But any tail-head combination, a 1/2 a chance. So there's more than one way to get the same thing. Does that make sense? Okay. All right. So that was the 3rd law, the law of independent assortment. All right. Then what we did is we looked at those laws, we started doing crosses, we started putting those together, and then we said, okay, let's look at pedigrees. Okay. And then there were 3 pedigrees I wanted you to look at. I wanted you to be able to identify whether the particular pedigree was an example of a dominant, a recessive, or a sex-linked recessive. The sex-linked ones will always be recessive. So let me give you some practice looking at some different pedigrees and we can analyze that. Oh, can you put a pin in that? Can I say one more thing first? You all right with that? Side note. I forgot to talk about this with independent assortment, right? The idea is you don't see independent assortment. That would be the exception to the independent assortment rule, just like we had exceptions to the dominant rule. The exception to the independent assortment rule is when genes are not on different chromosomes. If they're on the same chromosome, they're traveling together. They're not going to assort independently. So linked genes are the exception to the law of independent assortment. Linked genes are the exception to the law of independent assortment. All right. Now, what will break up those gene linkages?
Crossing over.
Yes, crossing over will break up those gene linkages. And you're going to probably have fewer crossing overs, right, than you would non-crossing overs. And then there was one other thing we talked about in it, that is the percentage of crossing over that you have. If I were to compare crossing over here with crossing over here, okay, what linkage would you be most likely, am I spelling that right? To break? Yeah, would it be ZG or would it be SR? ZG, because for the ZG to break it up, you can cross over here, here, and you'll break up that linkage, right? Whereas if you want to break up the ZR linkage, it has to be only right here. Does that make sense? Okay, so you know by how often the linkage is broken, how far apart they are. If it rarely gets broken up, that linkage is tight and it always travels together, you know they must be really what? Close together on a chromosome. If it happens frequently on that chromosome, you know they are really what? Far apart. Exactly. And they use that for gene mapping. Yes?
Does the green part where the two chromosomes come together?
That's the centromere, yes.
Does that have any effect on the
No.
maximum?
All right. Now, let's look at some different pedigrees and let's decide, okay? For every one of these, we're going to decide, yes or no, could this be an example of a dominant trait, a recessive trait, or an X-linked trait? That's what we want to decide, okay? So do you want to try on your own? First, check dominant trait. Could this be an inheritance pattern of a dominant trait? What do you think? Yes or no?
Yes.
I agree. Okay? The way I would do that, remember the trait is dark, right? It's filled in. That's what we're following. So if this is a dominant trait right here, I know they have to be at least what? Big A something. I don't know what. But they're at least a big A because they have the dominant trait, right? This individual here also has to be what? Big A something, right? I know Dad over here does not have this disease, right? Because it's not colored in. So what is his genotype?
Little a.
He is little a. So do I know this individual's genotype?
Yeah.
Yes. What is it?
Big A, little a.
Big A, little a. Okay? So yes, it will work for a dominant trait. Do you see how I did that? Okay, now let's try a recessive trait. We can use the same letters, but I'll use a different color to be helpful. So if you have the disease, what are you right here? Recessive trait.
Little a.
Yes. So little a. This one is?
Little a.
Okay. This one does not have the disease, so they have to be at least?
Big A.
Could they be harboring a little a? Yes. So will it work for a recessive trait? You see that? Yes. Yes, it will. I have one slide that has all three of these on here if you want that. Okay? So this will work for a recessive trait and it works for a dominant trait. Okay? Now, let's try a sex-linked trait. So the first thing I would do is I would just, what color have I done? I'll do is green. I know this is a girl, so what is she? XX. Mom is? And dad is? Okay. So, remember all of our diseases that we're going to look at are recessive. Okay? So I'm going to say they're not normal if they have the disease, so I'm going to put little n for not normal, right? Normal would be the dominant one. You okay with that? Okay, so mom has it, so she's X little n, X little n. Daughter would have it, they would have to be what?
X little n, little n.
Right. Now, they could get one of these bad X's from mom, but the other one has to come from dad. Does dad have the disease? He has to be then big N. So, would it work for an X-linked disease, this one?
No.
No, it does not. Does not work for X-linked. Okay? Because that cannot happen unless mom was sleeping around. Does that make sense? I don't want to, I'm not trying to besmirch mom. She's very wonderful. All right, ready for another one? Okay, let's do the same thing. Okay? Would it work for dominant? Would it work for recessive? Would it work for X-linked? Okay, what do you think about dominant? No, because we know dominant traits don't skip a generation. And for you to have it, you would have to have a big A here, right? No way to get that because both parents don't have the disease, because this is a dominant disease. We're just being hypothetical. We don't know what it is. We're geneticists. We're trying to analyze it. And we're saying if this were, no, a dominant disease is not inherited in this fashion. Okay, so it's not dominant. So no to the dominant. All right, what about a recessive disease? Yes, because this individual who has it would be what? Little a, okay? Could these parents be keepers, hiders of little a? Yes. Okay, so yes, it does work for recessive traits. Okay, now let's try sex-linked. What's your gut reaction on this for sex-linked? Let's write it out. First, let's put all their sexes on there, right? So here's what? Little girl, little boy? Little boy. Here's mom, yeah? Here's dad. Okay. Now, does this dad have the disease? Does this son have the disease? Yes. So I'm going to give him a little n because all the sex-linked diseases I give you are going to be recessive. Okay. Who did he get the Y from?
Dad.
Got it from dad. Okay, so that's cool. Could he have gotten this X from mom? Yes, but she doesn't have the disease. What does that tell you?
She has one big N.
Yes. One big N, one little n. Does that make sense? So will this work for sex-linked? Yes. You good on that? Questions, concerns? Want one more?
Yeah.
Okay, here you go. So we want to know, will this work for a dominant? Will it work for a recessive? Will it work for X-linked? What can you rule out easy peasy?
Dominant.
And how are you ruling out that? We don't even need to put any letters. Dominants don't skip generations. We're not even looking at you. Okay? We're done with you. Okay? How about a recessive disease?
Because one can have two parents.
Let's try. They have the disease. What are they? Little a, right? This mom, right? She could be big A. We don't know, right? The daughter has at least a... Yes, at least a big A because they don't have the disease. But what else does the daughter have?
Little A.
How do you know she has a little a?
Because
Her father only has little a's to give. So she could be big A, little a, yes. She marries this man. Does he have the disease? No. Remember, we're talking about a recessive disease now. So he has at least a big A. Could he be harboring a little a?
Yes.
Yes. And that's how you would make this son that is little a, little a. And then this girl, we don't know, but she has at least a big A. So does it work for sex-linked or recessive?
Yes.
Yes. We can say yes to recessive. Let's try sex-linked. Let's put some sex on here. Oh, that sounded terrible. Did I do right? Okay, so now let's put on some disease, okay? Does dad have the disease?
Yes.
Then he has a what? Little n, okay? Now, does mom have the disease?
No.
So what does she have?
At least one big N.
Right, but that's all I know, right? I don't know what her other letter is. I'm going to put a dash. Could they have had this little girl that didn't have the disease?
Yes.
Yeah, how?
If she had a little N.
not if she had a little N, but if she had who could she get a big N from? Her mom can give her a big N, X big N. We know what her dad gave her. That's all he had, one card to play. What did he give her? A little N. So she is a what? Carrier. We don't know anything about mom. All we know is she doesn't have a disease. We don't know if she's a carrier. We don't know. Okay? So this daughter is a carrier because her father had it and that's all he had to give. Okay, does this father, does he have the disease? No, so what is he? Big N. Okay, so here's a son. Who did he get the Y from? His dad, right? Who would he get his X from? Mom. What were the odds that she was going to give him a bad N?
50-50.
Yeah, 50-50. That seems like pretty high odds to me, which apparently she did, and that's why he has the disease. The daughter doesn't have the disease, but even if I didn't show you, I could ask you, do you think the daughter will have the disease based on these parents? What would you tell me? No. Why? Why would you tell me no? Yeah, dad only has a big N to give, and I don't even know what mom gave. She gave the big N or she gave the little N. So will that work for a sex-linked disease? Yes. Okay, and if you want, you have it on the video what they all do, but here would be if you want a picture, right? And if we were going to list, if you wanted like an answer key to it, right? This one right here will work for what? What diseases? Dominant and
sex-linked?
Not sex-linked.
No,
recessive. This one will work for recessive and sex-linked. And this one will work for recessive and sex-linked. Okay, and I'll move out of the way so you can have that too. All right, what did I have here? Oh, getting ready for chapter 23, quiz best bets. Okay, make sure you can understand practice problems 1 through 6. Okay, that's practice problem set 1, practice problem set 2. Where are the answers to those? Yeah, and if you have questions on those, I'm happy to go over any ones you want to have questions on. And then make sure you understand the testing yourself questions at the end of the chapter. They're on page 486 to 488, and the answers for that are in the back of your book. And then you need to do your additional objectives. Do you have any of the practice problems that you want to do? You had questions on? Good? Feeling strong? Yeah. Good review?
Yeah.
You know I love you?
Yes. Yeah.
Okay. All right. Have a good evening. Don't stay up too late. Do practice problems. That'll get you ready. I love you. Have a piece of toast.
Piece of toast.